Good afternoon to all. I am Gothaiammal, working as Assistant Professor from Department of Microbiology. Today we are going to discuss about the topic Giardia, lamblia. It is a parasite. Today we are know the Giardia, lamblia, morphology and life cycle and how prevent the organism from disease. First we know about the introduction. Giardia lamblia is a flagellated protozoan parasitic that inhabits the small intestine of humans and other mammals. It is one of the most common causes of intestinal parasitic infection worldwide, leading to a disease known as giardiasis. The infection is usually transmitted through ingestion of cyst present in contaminated water, food or via the fecal oral route. Then we know the morphology of Giardia lamblia. The morphology of protozoa consists of two main stages, that is the trophocyte and the cyst. The trophocyte is the active, motile and feeding stage during which the organism grows and multiple inside the host. It is a delicate and cannot survive long outside the host body. In contrast, the cyst is the dormant, non-motile and infective stage that is responsible for transmission from one host to another. The cyst form is resistant to harsh environmental condition and can survive outside the host, making it the main form found in contaminated food or water. Then we know the life cycle of Giardia lamblia. The life cycle begins when a host ingests mature ingest, which are the infective form, through the contaminated food, water or direct contact. Within about 30 minutes of reaching the duodenum, excitation occurs, releasing two trophocytes from each cyst. These trophocytes multiply by binary fission and colonize the duodenum and upper jejunum, where they feed by pinocytosis, often leading to malabsorption and diarrhea. Under unfavorable condition in the intestine, the trophocyte undergo encystation, mainly in the large intestine, forming mature cysts. These cysts are then excreted in the feces and can remain viable in soil and water for several weeks, thereby facilitating the infection of a new host. Then pathogenesis. Giardia infection begins when a person ingests cyst through contaminated food or water. Once in the duodenum, the cyst undergo excitation, releasing trophocytes. These trophocytes attach to the epithelial cells of the small intestine using their ventral sucking disc, causing villous atrophy and damage to breast border enzymes. This leads to malabsorption of nutrients and result in diarrhea, abdominal discomfort and nutritional deficiency. The host mount an immune response producing intestinal immunoglobulin A and recruiting inflammatory cells. Depending on the effectiveness of the immune response, the infection may be cleared or may persist as a chronic infection. Next, clinical manifestation of Giardia lamblia. Giardiasis is typically present with gastrointestinal symptoms such as diarrhea, that is watery, greasy and foul smelling, accompanied by abdominal cramps, bloating and excessive flatulence. Patients may also experience nausea and occasional vomiting. In chronic cases, malabsorption of fats, carbohydrates and vitamins can lead to weight loss, fatigue and lactose intolerance, while children may fail to thrive. The incubation period is usually 1 to 3 weeks and symptoms can last from a few days to several months if the infection becomes chronic. Then how to diagnose the Giardia lamblia? The laboratory diagnosis of Giardia lamblia infection is primarily based on stool examination. Trophocytes can be detected in fresh diarrheal stools using a direct wet mouth method, while cysts are identified in formed stools through the concentration techniques such as formalin ethyl acetate sedimentations. Stains like trichrome or iron hemotoxylin improve the visualization of the parasite. In addition, antigen detection test, that is ELISA or rapid immunochromatographic assays, it provides the sensitive and specific identification of Giardia in stool sample. Molecular methods such as PCR is nothing but it is the polymerase chain reaction, are used in special cases for precise detection, while procedure like duodenal aspirate or the string test are rarely employed when stool tests are inconclusive. Multiple stool samples are often required due to intermittent shedding of cyst. Then how to prevent safe water supply, drinking boiled or filtered water, avoid untreated water from lakes, rivers or wells, food hygiene, wash fruits and vegetables thoroughly, avoid eating raw or contaminated food. Personal hygiene, hand washing with soap, especially after using the toilet and before handling food. Teach children proper hygiene to prevent person-to-person transmission. Environmental measures, proper sewage disposal and sanitations, avoid fecal contamination of water sources. Next, how to control from giardiasis. Treatment of infected individuals with anti-giardial drugs. Example, metronidazole, diazonal, to reduce the transmission. Public health education on hygiene and safe water practices. Prevent outbreaks in daycare centers, schools and communities by monitoring and isolating cases. Then conclude the Giardia lamblia. It is a significant intestinal pathogen and controlling its spread is crucial to reducing the gastrointestinal illness, especially in regions with poor sanitations. Thank you.